proteins. Proteins are basically polymers of amino acids. And we have lots of different amino acids, about 20 or so. Some of them are essential, meaning that we can not make them in our body. We have to bring them in by diet or supplements. And then some of them are non-essential, meaning that our body can make them organically. And so basically these building blocks can be anywhere from a short peptide, which is about 10 to 20 amino acids linked in a chain, or they can be much more complex, up to about 100 amino acids linked in a chain. Okay, now the way we form these proteins is via a peptide bond. So we covalently bind the carboxyl group. Let's illustrate that here. So the carboxyl group on one amino acid with the amino group on another amino acid. And we basically have to release water. What type of reaction is this? If we're releasing water to form this peptide bond, what type of reaction is that? We saw a very similar phenomenon when we look at the formation of saccharides. Condensation, yeah, perfect. So condensation, because we're releasing water in order to form this covalent bond. And this is all that proteins are. One amino acid linked to another amino acid, to another. And they can, again, be as small as about 20 or so amino acids to much more than 50 up to 100 amino acids. We're going to look at some of the different structures that we can see. So on the primary structure, this is the first or most basic level of protein formation. This is the primary structure, and this is simply one amino acid covalently linked to another amino acid, right? Nothing complicated about this, and we form this peptide chain. Now, as we go on, we get to a little bit more complex of a structure. This is the secondary level of structure here. And what we have is a bond between the hydrogen on one amino group and the oxygen on the carboxyl group. And this allows us to form some more intricate, complex structures. Two examples here are the alpha helix, which is this helical-like formation, and then the beta-pleated sheet, which is this hairpin loop formation on the right. And so as you can see, we have the basic primary structure, which is one amino acid next to another, and then we have a secondary structure where they're forming more complicated configurations here. Let's move on to the tertiary structure. So this is where things get a little bit more complicated. We can have several different alpha helix or beta pleated sheets coming together to give us the tertiary level of structure. And from this level of structure, this is where the molecule becomes really sensitive to heat and to temperature, right? It can be denatured if we misfigure the formation of the protein at the tertiary level. This is why in excess acid, proteins are denatured. You lose this configuration, this conformation, which is the tertiary level and beyond. All right, and then finally, the quaternary structure is the most complicated or complex of the structures of proteins. We have quaternary structure where we can see multiple alpha helices or multiple beta pleated sheets. We can see a lot of different things happening here. And although this looks like one big jumbled mess, there's a lot of order here. There's a lot of organization, there's a lot of structure in this molecule. And even the slightest change, okay, even the slightest change can lead to pathology, disease, and even be fatal. Does anyone know what this molecule is? Anyone know what this specific molecule here is? It's a really important one in the body. It has 4 subunits, and it's responsible for transporting oxygen. Any thoughts? Really important for transporting oxygen. It's a globular protein, meaning that it's in this sort of jumbled ball, almost like if you crushed up a piece of paper. So it's hemoglobin, right? Hemoglobin. And as we can see, hemoglobin, even the slightest change in the structure of hemoglobin leads to sickle cell disease. It's a single miscoding of a single nucleic acid that codes for this protein. And just the smallest or slightest misformation can lead to fatality, can lead to disease, as we see with sickle cell. So it's really important for proteins to hold their quaternary structure, and they're really sensitive to being denatured, to being dysfunctional if we shift or change this structure. Okay. All right. Lastly, are nucleic acids. Nucleic acids, now these are the building blocks of our DNA. They also form RNA, which can be then coded to DNA or proteins. And then nucleic acids are basically a phosphate group on the left here that are bound to a 5-carbohydrate ring, which can be one of two types. It can be a ribose, or it can be a deoxyribose, which has one less oxygen. So it can be a ribose or a deoxyribose. That'll make it DNA or RNA. And then lastly, we have a nitrogenous base that's added on to this molecule. So the nitrogenous base can be one of several flavors here on the right. They can be pyrimidines, which have a single nitrogen ring, or they can be purines, which have 2 rings, right? So based upon this, we can get cytosine, thymine, uracil, which are pyrimidines, or we can get adenine and guanine, which are our purines. So these are basically the building blocks for our DNA. We know our DNA is really, really important in terms of coding for everything from the way we look and our physical characteristics to our behavioral characteristics, our genetics, and things like that. So really, really important molecules. They're also important in terms of energy transfer. So we're going to talk a lot about adenosine, monophosphate, diphosphate, and triphosphate, which are AMP, ADP, and ATP. And these are basically the cellular currency in the body. This is how the cell sort of pays for or uses this energy to do cellular work. So this is how we make proteins. This is how we export things. This is how we shift around molecules in the cell. And we need this energetic currency in order to do that. So it's a really important part of that process. There's also NAD and FAD, so nicotinamide, adenine dinucleotide, which is NAD, and then flavin, adenine dinucleotide, which is FAD, are important carrier molecules for electrons. We're going to talk a lot about how these can shuttle electrons in and out of the mitochondria and help with oxidative phosphorylation, which is the primary source of ATP in our body. Okay, so really important in terms of the cell's energetic needs are these forms of nucleotides and nucleic acids. We also have the ring form of these nucleotides. This is called cyclic AMP and cyclic GMP. These are really important in terms of signaling and messengering in the cell. We'll also see this as we go through the semester, but basically there's a sequence of events within cells where one molecule tells another to do another and another. And that sequence of events, which we call the signal transduction cascade, is brought about usually by cyclic AMP or cyclic GMP. So these are really important messenger molecules that can help us relay certain messages to tell the cell to do something versus not doing something, okay? So 3 important functions of our nucleotides: DNA, energy transfer, and then messenger molecules.